All right, ladies and gentlemen, so welcome back. We are going to really start getting into some of the basic components to cellular respiration. And so when we look at cellular respiration as an overall process, it is aerobic. And it is going to take that glucose, use oxygen, and be able to produce copious amounts of energy. But um, we are going to break cellular respiration down into basically three major processes. You've got your glycolysis, which is going to happen here, your citric acid cycle, and then oxidative phosphorylation, which is going to include two sub processes called your electron transport or electron transport chain, as well as chemoosmosis. Um, so when we look at individual processes, not all of these are going to necessarily be requiring the oxygen. So not every process of cellular respiration is aerobic. Some of them will be anaerobic, but overall the process requires the oxygen. We are also going to trace glucose through this whole process for a couple of reasons. One, it gives you the overall process, every step that could potentially happen really starts off with that glucose. That being said, keep in mind that your body is capable of feeding in fats and proteins into this process. They're all capable of being utilized as fuel, but using glucose as the mechanism to teach you this process gives us the ability to talk through all of the steps. Um, and number two, your body is designed evolutionarily to use that glucose first, use those sugars, use those carbohydrates as its main fuel source. Um, if you think about it, go back to hunter and gatherer days, kind of in our early evolution as a human species, and we weren't evolving to use fats as our main food source. Proteins, same deal. Your body was evolving at that point where you may have gone days without access to food. And so we wanted to be able to store up any excess fuel in those fat stores so that when you went for a few days without having access to regular food, you didn't starve to death. Your body could go after those fats. So your metabolism is evolutionarily designed to take advantage of those sugars when those sugars are in excess, convert them back to fats. The fats that you eat, we store those immediately as well, um, and then only go after those fat sources when we're in an emergency. We're talking now, modern times, where most people have regular access to food, and that's not so much of a problem. And so it's going to require us to be mindful of caloric intake, exercise, the types of foods we're eating, in order to force our bodies to access that fat metabolism or to force our bodies to prevent us from storing excess sugars as fats because we're basically fighting our own evolutionary history. Proteins, the same deal. Your body doesn't want to use proteins as its main energy source because your cells are driven by their structure and their function. All of that's protein based. Enzymes, transport proteins, regulatory proteins, you name it, it's protein. And the only way your body can build those proteins is for you to consume proteins, break them down into the component amino acids, and then for your cells to use protein synthesis to rearrange those amino acids to create the proteins that you need. So your body, once again, doesn't want to go using those proteins as its major source of fuel because we need them. We need them in order to function. So that's the other reason we're going to trace glucose. So not only is it just an ability for us to teach you all of the component steps, because glucose really is what feeds in first, the fats and the proteins will feed into other places in this process, but it's also evolutionarily what your body wants to use as its main source of fuel. So again, I mentioned that cellular respiration, we're going to break it down into three major components. Your glycolysis is going to take that glucose and break it down into these two pyruvate molecules. So one of the things you're going to find is helpful as we're working through what's happening is to be able to trace what happened to the carbons from that original glucose. So hopefully at this point you know that glucose is C6H12O6, right? It's a 6-carbon molecule. So when we look at glycolysis, think about what that term actually means. Maybe you can identify some of the prefixes and suffixes, the Latin roots that are here. Glyco literally translates to sugar, and lyse means to break. So we're going to quite literally take that 6-carbon glucose and break it. In fact, we're going to break it in half. And so each of those pyruvate molecules gets 3 of those original glucoses. So we're going to take a 6-carbon glucose and break it down into a pyruvate molecule that is 3 carbons. We're going to make 2 of them in order to account for the 6 carbons in the glucose. So 2 3-carbon pyruvates. We're going to then take those pyruvates and now actually end up in your mitochondria. So glycolysis happens out in your cytoplasm or your cytosol. Citric acid cycle and oxidative phosphorylation both happen within components of your mitochondria. So that pyruvate gets fed into that citric acid cycle. Heads up, you may have learned it in the past or you may come across it again as the Krebs cycle. It's the same thing. Krebs is just the guy who discovered it, so he gets credit for the cycle in that respect. Citric acid is also the first molecule that gets produced in the cycle, and so that's where that term citric acid cycle comes in. Kind of depends on who you talk to. Most of the higher level textbooks, professors, I would imagine, probably stick to that citric acid cycle. But a lot of the more high school level textbooks tend to call it the Krebs cycle, so I throw both out there in case you come across either of them. It's the same thing. What's that cycle going to do, at least for our purposes at this moment? We'll go into more detail later, but it's going to take that glucose and completely break it apart. So by the time we get through the citric acid cycle, those carbons have gotten dispersed into all sorts of places, and it's going to be kind of hard to trace the carbons beyond that citric acid cycle. So what else is going to be helpful to trace? Well, not only where the carbons go, but then where do the electrons and the energy go? So when we look at both your glycolysis and citric acid cycle, that full breakdown of that glucose molecule is going to be allowing us to shuffle electrons and hydrogens around to create these electron carriers, right? So there's that huge redox component to this. We're going to finish those major redox reactions by creating NADH and FADH2. Those electron carriers are taking electrons and hydrogens and shuffling them over to this final step here, and that's the oxidative phosphorylation, which is going to include our electron transport chain. So that's where we're going to really utilize those electrons and those hydrogens to create a hydrogen gradient. And we're actually going to create that gradient between that inner membrane space here and the matrix of your mitochondria, the innermost component of your mitochondria. And we're going to use that active transport system, those pumps, to push those hydrogens into that gradient, force it to be there, create it to be there, and then allow a passive transport process via chemoosmosis to allow those hydrogens to diffuse back into the matrix and actually produce a huge amount of ATP as a result. So we're going to use that concentration gradient to then allow us to co-transport and create ATP. So when we look at the types of phosphorylation that are occurring, how do we get the ATP that exists? Well, most of the ATP is going to come from that oxidative phosphorylation process. So we'll talk more detail about that later. But you do get a little bit of energy from glycolysis and a little bit of energy from the citric acid cycle. But rather than oxidative phosphorylation, where we're going to use oxygen as a major means of creating that ATP, that substrate level phosphorylation just gets us a couple of pieces. So oxidative phosphorylation, right, this is going to be part of that last step. It's going to produce the vast majority of your ATP, completely powered by those redox reactions that we were doing throughout this whole process, creating those electron carriers, moving them here. And almost about 90% of the ATP generated by cellular respiration occurs through oxidative phosphorylation. Again, we'll talk in a little bit more detail exactly how that process works as we talk through more details about these processes. The rest of that ATP, though, comes from the substrate level phosphorylation. And this is by no means the amount of efficiency that we can get from that oxidative process, because it's really one ATP being produced by the enzyme at a time. So when we look at the substrate level phosphorylation that comes out of glycolysis in the citric acid cycle, both of those processes are each able to produce only 2 ATPs apiece. We get to your oxidative phosphorylation and we're going to get somewhere on the order of 30, 32, 34 ATPs. So it's a huge difference. But if we look, the basic idea here is we take an enzyme, substrate that has been phosphorylated, we fit an ADP and that phosphorylated substrate into its active site. Enzyme does its job by catalyzing the break of this phosphate and the addition of that phosphate being added back to that ADP. We get a product that gets released and we get a little ATP out of it as a result.